In the previous video, we talked broadly about the divisions of the nervous system. In this video, we'll take a deeper dive into how neurons, the cells of the nervous system, actually work to help your body send and receive messages. The nervous system is comprised of two basic types of cells, glial cells and neurons. Glial cells play a support role to neurons. They provide insulation, transport nutrients and waste products, and also help with immune responses. Neurons are your body's information processors. They send and receive information. Without neurons, your brain wouldn't be able to function and it wouldn't be able to communicate with the rest of your body. So neurons play a pretty important role. In fact, at birth, you have over 100 billion of them. Let's first look at the basic structure of a neuron cell, and then we'll talk about neuronal communication. The cell body of the neuron, which contains the nucleus, is called the soma. Attached to one end of the cell body are long branches called dendrites. Dendrites are responsible for receiving information from other nearby neurons. On the other end of the cell body is a long fiber called an axon. Neurons transmit or send information by sending electrical signals down the length of the axon. Axons can range in length from a fraction of an inch to several feet. In some nerve cells, glial cells form a fatty substance known as a myelin sheath, which coats the axon and acts as an insulator, thus increasing the speed at which the electrical signal can travel. Multiple sclerosis is an autoimmune disorder that involves a large-scale loss of this myelin sheath. The result is that the electrical signal can't travel as effectively, meaning that neurons can no longer communicate as efficiently. This can lead to a number of symptoms including dizziness, fatigue, and loss of motor control. At the end of the axon are several terminal buttons that contain neurotransmitters, which we'll talk about more in a minute. When an electrical signal travels down the length of the axon, it can trigger the release of these neurotransmitters at the terminal buttons. Neurons communicate via an electrochemical process. This means that it involves both an electrical and a chemical message. The electrical signal that travels down the length of the axon is called an action potential. In order to understand how an action potential works, however, we first need to understand what's going on when the neuron is not actively sending an electrical signal. This is called the neuron's resting potential. Even though the neuron is at rest, in the sense that it's not actively firing a signal, it is actually kind of an unpleasant state, at least for the neuron. This is because the neuron intentionally creates a state of tension. In this resting state, sodium ions, shown here in dark blue, are at higher concentrations outside versus inside the cell. Sodium is positively charged, and the inside of the cell is slightly negatively charged. Remember that opposites attract, so in this resting state, sodium ions would really like to get inside that cell if they could, both because of the difference in electrical charge and the difference in concentration. However, during the resting potential, the cell membrane of the neuron actually prevents the sodium ions from entering the cell. So this creates the tension. When the cell is at rest, sodium would really like to get inside that cell, but it can't. Essentially, this is the same idea of stretching a rubber band back. The rubber band is still, but it is filled with potential energy. As soon as you release it, the rubber band will fly across the room. In the same way, the resting potential gets the neuron ready for action. This action starts when the neuron receives signals from the dendrites. Next, small pores or gates open on the neuronal membrane, which finally allow all those positively charged sodium ions to rush into the cell. With a sudden influx of positive ions, the internal charge of the cell becomes more positive. If that internal charge reaches a certain level, called the threshold of excitation, then the neuron becomes active and this starts the action potential. If the threshold for excitation is reached, then an electrical signal will move down the length of the axon. This signal propagates or travels down the axon by jumping between gaps in the myelin sheath called nodes of Ranvier. This moves the action potential all the way down the axon until reaching the terminal buttons. After an action potential, the neuron enters a refractory state while it essentially resets, pumping sodium ions back outside of the cell. During this refractory state, the neuron is incapable of starting another action potential. The action potential is an all or nothing event. If the threshold is reached, an action potential will occur. If not, it won't. It's kind of like taking a class pass-fail. There's only two possible outcomes. Also, if an action potential occurs, it will always continue at full strength until it reaches the end of the axon. There's no stopping it halfway. You can check out the supplemental videos on Canvas for more information on how resting and action potentials work. When the action potential arrives at the terminal buttons, this starts the chemical part of the neuronal communication process. Specifically, the action potential tells the neuron to release chemicals called neurotransmitters. After being released by the terminal buttons, these neurotransmitters then travel across a very small gap called the synaptic cleft, where they bind with special corresponding receptors on the dendrite of an adjacent neuron. For an example, here's a graphic showing how multiple neurons might communicate. If neuron number 1 has an action potential, it will release neurotransmitters at synapse number 1. If enough neurotransmitters then subsequently bind onto the dendrites of neuron number 2, then that will start an action potential in neuron number 2, and so on. Some neurotransmitters are excitatory neurotransmitters. That is, they make it more likely that the postsynaptic neuron will have an action potential, as in the example I just described. However, other neurotransmitters are inhibitory. That means they make it less likely that the postsynaptic neuron will have an action potential. For example, again looking at the graphic here, if inhibitory neurotransmitters are released at synapse number 1, and if enough of them bind to the dendrites of neuron number 2, then that will actually make it less likely that neuron number 2 will start an action potential. In addition to being either excitatory or inhibitory, different neurotransmitters also appear to be associated with different specific functions. For example, you may have already heard of the neurotransmitter serotonin before. Serotonin is involved in regulating sleep, eating, and mood. Another neurotransmitter, dopamine, appears to be involved in reward processing. Other common neurotransmitters include acetylcholine, which is involved in memory, endorphins, which help with pain relief, glutamate, a major excitatory neurotransmitter, and GABA, a major inhibitory neurotransmitter. Each of the different neurotransmitters have a different shape that is designed to fit only certain types of receptors on the postsynaptic neuron. For example, a GABA neurotransmitter couldn't bind to a serotonin receptor, and vice versa. Some substances, either natural or synthetic, have a chemical structure that is very similar to certain neurotransmitters. For example, caffeine has a very similar structure to adenosine, an inhibitory neurotransmitter that slows your body down and can lead to you feeling sleepy. Because of their similar shape, caffeine can actually fit into the receptor sites for adenosine, effectively blocking those receptors from being accessed. This means that adenosine can no longer effectively do its job, which is to make you feel sleepy, because its receptor sites are being blocked by the caffeine molecule. The end result is that you end up feeling more alert, at least for a little bit. In the long run, however, this actually leads your brain to compensate by creating more adenosine receptor sites, eventually leading to caffeine tolerance, where you need to keep consuming more and more caffeine in order for it to have the same kind of effect. Other kinds of drugs work via a similar mechanism. For example, some antipsychotic medications work by blocking certain dopamine receptors in the brain, thereby reducing the overall activity of dopamine in those certain brain regions. To learn a little bit more about how caffeine works, you can check out the supplemental video in Canvas. In this video, we covered the different parts of neuron cells. We also discussed how neurons send and receive information via an electrochemical process. For the exam, you should be able to identify the different parts of a neuron cell, define and describe the resting potential of a neuron, describe how the process of an action potential works, describe how neurotransmitters are used to communicate between neurons, and identify the functions of the neurotransmitters we talked about in this video.